Hey, you guys, I hope you're staying in and you're staying well. Office hours went well yesterday over Zoom, so I will make myself available to do that on Mondays and Fridays, which I should be able to do provided I get my other lectures out of the way and posted. And I'll post an announcement to Canvas regarding next office hours, probably Friday. We'll spend the rest of the course talking about higher brain function within the cerebral cortex. So this lecture will be kind of a bridge lecture. We've had a couple of bridge lectures earlier in the course where I'll remind you of some things that we've previously discussed and talk about some things that we will be discussing in coming lectures, and in between share a little bit of new information. So that's our trajectory for the rest of the course. And those of you, particularly in behavioral neuroscience, may recognize the gentleman over here on the right, Henry Mollison, or patient HM, as he was referred to in the literature when he was still alive. We'll talk about patient HM. He underwent bilateral removal of his hippocampi when he was a young man, 27, in order to cure debilitating tonic-clonic, formerly known as grand mal seizures. And this surgery effectively cured his seizures, but came with the horrific side effect of leaving him unable to form any new memories. So he would essentially live a few minutes at a time before everything would reset and start over in terms of his memory. So we'll talk about patient HM, and I'll have you read a couple of papers once you've listened to the lecture and worked through the slides. Okay, so we will start off talking about a little bit of neuroanatomy. In other words, we'll look at the layers of the cerebral cortex and the cryostructure. So cryostructure is looking at the different cell types that are present in different layers using different types of staining techniques. We'll talk about Henry Mollison, pathophysiology, the diagnosis of epilepsy, his surgery, and what the side effects were in terms of learning and memory. And then we'll differentiate between a few different types of memory. I'll give you some definitions and give some examples. And then we'll talk again. We've already started talking about learning and memory, and you should be able to discuss LTP and LTD and everything we've learned up until now. And we'll revisit the role of the hippocampus and the entorhinal cortex. And then again, related to HM, patient HM, the effects of loss of the medial temporal lobe, MTL here is medial temporal lobe, on anterograde and retrograde amnesia. Okay, so we saw this slide in a previous lecture, so we'll be spending our time talking about the medial temporal lobe and the limbic system today, and their role in learning and memory, particularly short-term, episodic, and fact-based memories. And then next time we'll talk about the prefrontal cortex, which is important for decision-making and goal-directed behavior. So that'll be next time. But a lot of the prefrontal cortex develops during adolescence, and so a lot of changes happening in the prefrontal cortex, which isn't fully developed until adulthood, but we'll leave that for next time. Okay, so 90% of the cortex is neocortex, meaning in terms of evolution, it evolved more recently. So if it's not neocortex, then it's paleocortex or archicortex, which are more evolutionarily ancient regions of the brain. So the different types of staining here. So a Golgi stain sort of stains everything. It stains the entire cell. A Nissl stain stains the cell bodies. So you can see the cell bodies highlighted here. And a Weigart stain is for myelin. So you can see myelinated axons. And I'm probably not going to test you on different types of stains and how they work, but you can appreciate that we can learn a lot about the cryoarchitecture or cryostructure of these different layers using different types of stains. And hopefully it makes sense that what you're seeing here is axon tracks. Here you're seeing cell bodies, and here you're kind of seeing entire cells based on these three different stains. Your job is to know what is present in each layer. So we've said that throughout the course, the neocortex is a 6-layered structure. And in these two diagrams, you can see a depiction of what is present in each of those 6 layers. Layer 1 largely axon terminals. Layer 2, stellate cells. Layer 3, pyramidal cells, which are largely excitatory. Layer 4, more stellate cells. Layer 5, pyramidal cells, and efferents going to subcortical regions, perhaps the thalamus, basal ganglia, or otherwise. And then layer 6, spindle-shaped fusiform cells, pyramidal cells, and cells that are important for connections between the cortex and the thalamus. So those would be corticothalamic or thalamocortical efferents. So if you know in general what's present in each layer, that's fine. And I've made some additional notes here for you on the next slide. So I won't read this slide to you. You can read it yourself, but I should have paraphrased what was on the previous slide when I was talking you through it. These are the 6 layers and what you can find in each of those 6 layers. And so if you know this in general, that's fine, and I'll try and be clear about where the layers are particularly important. I'll point that out when we get to it. The neocortex, remember that's evolutionarily newer, has 6 layers. Archicortex, which is evolutionarily more ancient, has 3 layers, and paleocortex has 4 or 5, depending on which specific region you're looking at. So key takeaway, 6-layered structure of the neocortex. So the hippocampus is archicortex. Okay, so that would have a smaller number of layers. If you wanted to see the hippocampus, you would need to dissect the brain. It can only be viewed via dissection because it's folded into the temporal lobe. And the hippocampal formation includes a few different structures, many of which we've seen before. The hippocampus proper, where the hippocampus proper is subdivided into CA1, CA2, CA3, CA4, and we've looked at some of this circuitry previously. The hippocampal formation also contains the subiculum, and it contains the dentate gyrus. Dentate gyrus is up here, and the entorhinal cortex. And so the subiculum is an important structure because it connects the hippocampus proper to the entorhinal cortex. And remember, the entorhinal cortex is important for connections between the hippocampus and higher structures. So the 3 layers that are present here, if you go back a couple of slides and look at layer 1, layer 4, and layer 5 of the neocortex, the 3 layers that are present here are anatomically similar. Same types of cells are present as in layer 1, layer 4, and layer 5 of the neocortex. So main input to the hippocampus proper and the hippocampal formation is from the entorhinal cortex. And a number of you have asked in office hours about the exact circuitry between CA1 and CA3 and the dentate gyrus and the entorhinal cortex. And there's no one specific pathway that you need to memorize. There's all kinds of circuitry here, reciprocal connections between the hippocampus proper and the entorhinal cortex, and also crosstalk and pathways that connect the contralateral hippocampal formation. So there's no one pathway you need to memorize, but you should certainly know that all of these anatomical regions are part of the hippocampal formation. You should know that the subiculum connects the hippocampus proper with the entorhinal cortex, and you should know that the entorhinal cortex is an important bridge between the hippocampus and higher brain structures. Okay? And if you know that for now, that's good. And if and when we get to any specific circuitry or pathways or connections that you need to know, I'll be sure to point those out. All right, so Henry Mollison, and if you've got some time on your hands, this is an excellent book. Luc Didrich is the grandson of the surgeon who operated on Henry Mollison. And so he wrote this book about essentially the Henry Mollison's life before, during, and after the surgery. And he also wrote about his grandfather, who was a prolific and brilliant neurosurgeon, but he also arguably crossed some very strong ethical lines with some of the surgeries that he did. And I'm going to have you watch a video that'll explain that a little more. In any case, awesome book. My copy is on loan, but you can pick it up if you want to learn more about all of this. So you can look at this cartoon. This would be a normal brain, and this would be Henry Mollison's brain after the surgery. So bilateral, both sides removal of the hippocampi, and basically large fist-sized chunks of the medial temporal lobe is what the surgeon, whose name was William Scoville, that's what he removed. And so the reason that was done as an experimental surgery at the time was because after a bicycle accident when he was age 7, Henry Mollison developed seizures, and the seizures got progressively worse through his teenage years and early adulthood, to the point where he would have multiple tonic, clonic, or grand mal seizures every day. So you could imagine how that would impact school, learning, work, relationships, holding down a job, and basically any other aspect of life that you could possibly conceive. And so when he was 27, he underwent this experimental surgery by the surgeon that he had been referred to, William Scoville, and had both hippocampi removed. And that effectively cured seizures. However, he didn't suffer from retrograde amnesia. His past memories were intact. He could remember most everything up until just before the surgery. However, he had anterograde amnesia, meaning he no longer had the ability to form new memories. So imagine living a few minutes at a time and then not being able to remember anything that happened a few minutes ago, and having life kind of reset in 60-second intervals, or maybe intervals of a few minutes. And so the studies of HM after the surgery led to a major increase in our knowledge about brain function and anatomy and learning and memory. After the surgery, HM's personality was not changed. Next time, we'll talk about Phineas Gage, who suffered a different type of brain injury or brain damage, and his personality was dramatically changed. So you've likely heard of Phineas Gage somewhere along the way. This is not what happened in HM. There was no change in his personality. His personality was the same, and his IQ actually increased. Not because removing fist-sized chunks of the brain will make you smarter, but imagine the seizures, right? The multiple tonic-clonic seizures per day. In the absence of seizures, he could focus. He could learn, at least in the very short term. So his IQ increased, probably because he could concentrate in the absence of the seizures. He couldn't follow the plot of a TV show because, of course, everything would reset for him several times during an episode. But he was really good at crosswords. And studies showed that he could even modify pre-existing memories. So prior to HM's surgery, when he was growing up, polio would have been rampant, right? Polio would have been a devastating part of HM's childhood, and vaccinations had not yet been put into place. Vaccination developed by Jonas Salk was about the mid-1950s after HM's surgery. But because he had pre-existing knowledge of polio, because he grew up knowing about it, he was able to modify those pre-existing memories to include Jonas Salk. So that was interesting, modifying old memories. However, HM was living proof that the ability to form new fact-based and event-based memories relies on the medial temporal lobe, because that's what he had removed, right? Large chunks of his medial temporal lobe. However, overall cognitive function, i.e. crosswords and IQ and general knowledge, and motor memory don't. He was able to learn new motor skills. He might not remember that he had learned them, but motor memory don't rely on the medial temporal lobe. It's specifically fact and event-based memories that do rely on the medial temporal lobe. Okay, and so you can read this quote. It's pretty powerful and devastating at the same time. I mean, imagine what it would be like to be 27 and having everything reset every few minutes. And so then HM became a study subject, and he was involved in so much research, and there were so many publications generated, and we learned arguably the single most valuable study subject in neuroscience, period, arguably. But imagine, he would interact with the same people, the same nurses and doctors and researchers every day, but each morning or each afternoon when he saw them, he had no idea he'd interacted with them before. So he remained intelligent, perceptive, but of course he couldn't hold down a job or live on his own if everything is being reset every 60 or 120 seconds. So his life, the rest of his life, was a series of inconsistent, incoherent, non-threaded, isolated moments. And so it's pretty devastating if you take a minute to think about it. I will post this video. If we were in lecture, I'd put it up for you, but I'm going to have you read an article. It's National Geographic, and the first little blurb of it is here, and it talks about the surgeon who was responsible for HM's brain after his death. And so it was very carefully preserved over a period of months in a glucose solution, and then it was cryo-frozen, and then it was sliced into about 2,000 micrometer, or on the order of micrometer thin slices, and then it was uploaded. And you can go online and you can access HM's brain, which is pretty powerful and devastating at the same time. So I'll post a video of how the cryo slicing of his brain was done. But the surgeon or the researcher that was responsible for the slicing, and I'd tell you a story. I had an opportunity to go to Japan and do some of this type of work, not with brain tissue, but with eyes, right? You've deduced by now I spent a lot of time studying opsins and the retina. So we would do this type of cryo-freezing with eyes and do these very careful slices and then do similar types of imaging. So I want to show you how painfully painstaking this technique of making cryo slices is. So this video, and I'll post the link for you, will show you how painstaking. And imagine making 2,000 or 2,500 slices. The researcher who was doing this had a whole team of people that were there just to keep him awake and keep him focused and bring him coffee. And they had code words if he started to nod off, and so on. And you'll learn a little bit about that in the article. So I'll let you watch the video. I'll let you read the article, and then we can have a little more discussion about this. All right, so hippocampus, you know by now, the hippocampus is most critical for long-term memory, and particularly moving short-term memories into long-term memory. So the hippocampus is where memories are initiated. It's where memories are initially formed, and from the hippocampus, they are then stored in long-term memory. That involves the entorhinal cortex. So that's the interface between the hippocampus and the cerebral cortex. So there's different types of memory, and those of you that have taken some psychology, or those of you that are in behavioral neuroscience will know these different types of memory, but those of you that are in kin or physiology may not. So let's all be clear on different types of memory that involve the hippocampus and the entorhinal cortex. So an episodic memory is something that you experienced. You had to be there. If you're going to have an episodic memory about something, you might remember your 16th birthday party, right? You certainly remember your high school graduation, right? It was an event, you were there, and that would be an episodic memory. A semantic memory is something that's fact-based and you could communicate to me, right? Any of the information you've learned in this course, for example, would be fact-based memory. If you were to describe LTP, or the primary mechanism underlying LTP, that would be an example of semantic memory. You might not remember on which day in which lecture we talked about that, but you could tell me about it. Another example would be the grandchildren of Queen Elizabeth, right? You might not remember exactly where and from whom you learned about Prince William and Prince Harry, but you know that they are the grandchildren of Queen Elizabeth, right? And you probably know who their spouses are, and you may even know who their children are, depending on how much you follow the royal family. But that would be an example. And if you don't follow the royal family in Great Britain, you could extrapolate that to knowledge of perhaps another regime or group of important people. So things that you know, you don't necessarily remember exactly when and from whom you learned it, but you know it. So fact-based memory is semantic. Declarative is more of a general term, and these are memories that you could tell me about. You could verbalize them. So both episodic and semantic memories could also be declarative. Non-declarative are memories that are less consciously accessible and not so easy to verbalize. You can all tie your shoes, but if you had to describe to me the exact movements your fingers and your thumbs needed to make to tie or untie a shoe, that would be more difficult for you to do, right? Similarly, if you had to tell me exactly what I needed to do to balance on a bike and ride it, that would be more difficult. You can do it, but it's more of a non-declarative, less accessible type of memory. And then explicit and implicit, being aware of what you know versus unaware of what you know. And a good example is new skills that Henry Mollison learned after his surgery. And an example was he learned how to, how can I best explain this? Imagine you're looking at a mirror and you're looking at text being inverted or backwards on the mirror, and you have to read the text in the forward direction when the text is presented in the opposite direction. Okay, so you're looking in a mirror, somebody's holding up a card with a word behind you, and you see the word cat written backwards, right? It's TAC instead of CAT, and you learn how to read the word backwards, right? You can invert it. So HM learned how to do that after his surgery, but he wouldn't have been able to tell you that he had learned that new skill. So that would be an example of something that's implicit, right? Explicit is you know. Implicit is you know, but you don't know that you know. And that would be an example of something that HM had learned, but he didn't know that he had learned, which is therefore an implicit memory. I hope that makes sense. Okay, we've seen this slide before, some circuitry. Don't worry about this circuitry. I asked you about this previously when I asked you on an exam to talk about the paired pulse protocol as a mechanism to induce LTP. But again, I'm stressing you don't need to memorize this exact circuitry shown on this slide. This is simply a reminder that if you stimulate the entorhinal cortex does project through the dentate gyrus, and we looked at that circuitry previously, and that can be used to induce LTP. Okay? And we saw previously an experiment that you can do, the paired pulse protocol, where with time you will increase the amplitude of EPSPs, which is consistent with LTP and consistent with Hebbian principles. And so bilateral removal of the MTL or medial temporal lobes, including the hippocampi in HM, eliminated the circuitry. Don't worry exactly about the circuitry shown on this specific slide, but the circuitry and the neurophysiological mechanism for the formation of new memories was eliminated. So he could no longer make and store what would initially be short-term and then converted into or stored as consolidated into long-term memories. All right, we've talked about, earlier in the course, we talked about localizationism versus connectionism, and we said that both are important, and the balance between the two can change throughout life, from childhood into adolescence, into adulthood, and so on. So just like there was the Human Genome Project, right, which aimed to decode all of the base pairs and successfully did decode all of the base pairs in the human genome, we now have a Human Connectome Project that is aiming to kind of similarly map all of the neural pathways that underlie brain function. And I want to stress that there's a whole lot that we don't know about the connections between different regions of the brain. But certainly when completed, the Human Genome Project will produce very powerful data that will facilitate studies of all different kinds of brain conditions, brain disease, and neurological disease like Alzheimer's, like schizophrenia, and others. So techniques that are being used in efforts towards mapping the human connectome include positron emission tomography, functional magnetic resonance imaging, and I'm going to have to Google what fMRI, or let you Google what fMRI, but that's just a little more advanced than fMRI. And those are not techniques I'm going to ask you about in this course. This is just FYI and for interest. And so I'll post this article. This one's not required. Just if you're interested in things like mapping and the human connectome is interesting, then here's something that you can read. But otherwise, this is an FYI slide, not a you need to know it slide. In terms of plasticity, right, I want to stress plasticity and how if one part of the brain becomes damaged through stroke, through lesion, through some kind of damage, other regions of the brain can partially compensate. I told you a story about Trevor Green, the Canadian journalist and soldier who was attacked in Afghanistan and regained his ability to walk. That's an extreme demonstration of plasticity. And here's another one. So this paper is, again, I'll post it for you for interest, but there are a number of individuals in the population who appear not to have an olfactory bulb. I'm sorry, an olfactory cortex. No, an olfactory bulb. Sorry, I'm reading this as I'm talking. These individuals do not have an olfactory bulb, okay? Somehow they're still able to smell. So we should remember the anatomy of this pathway because we talked about this in recent history. So if an individual is lacking an olfactory bulb, that olfactory pathway is completely interrupted. So where is the olfactory information going? And it appears that even in the absence of an olfactory bulb, individuals can smell just fine. So that's another demonstration of plasticity. So that one's for interest. If you have a little extra time on your hands, you can read that one. Connectome. So this is recent developments in the Human Connectome project, and an image depicting some of the mapping. And here I want to remind you that earlier in the course we talked about Brodmann areas, which are anatomical divisions. And hopefully you can appreciate how something like a connectome, which is a functional mapping of the brain, is much more useful than perhaps Brodmann areas. So this might redefine how textbooks are written and how regions of the brain are referred to in the future once this project is complete. Okay, and then we saw this slide also previously. I just pulled it from an earlier lecture, and we were talking about this in the context of fear learning in mice, or fear conditioning. And in the short term, after learning, within a day, most of the associations involve the hippocampus. But in the days after learning, it's no longer the hippocampus. It's the cerebral cortex and the thalamus, the entorhinal cortex, acting as a bridge between the hippocampus and these higher regions. And so the fact that HM no longer had hippocampi, right, bilateral removal of the medial temporal lobes, that explains why he had anterograde but not retrograde amnesia. His old memories, the memories that he had in place before the surgery, those were intact. They were already stored. They were already involving the cerebral cortex and the thalamus. Removing the hippocampi wouldn't do anything to those memories. So he didn't have retrograde amnesia. But it also explains why he had anterograde amnesia, and it highlights the importance of the hippocampus and the medial temporal lobe in general in the formation of new short-term memories and the storage of new long-term memories. All right, so if we were in lecture, we would watch this together. I would play it in lecture. This is an interview with Luke Dietrich, that wrote the book that I introduced at the beginning, and who is the grandson of William Scoville, who was the neurosurgeon who operated on Henry Mollison. So I'll let you watch this, and do be sure to pick up on the nuance that William Scoville lobotomized his own wife. Okay? He was a prolific lobotomist. And so a lobotomy, for those that don't know, the bone at the top of your eye socket is quite thin. So you can take a sharp probe, and you can fairly easily pierce the upper region of an individual's eye socket. And above that region is your prefrontal cortex, where things like personality are regions of the brain that are important for personality. And so a lobotomist will use some type of tool to simply interrupt, mess up, disrupt the prefrontal cortex, and possibly simply by mashing it around a little bit with a sharp tool. And that was used as a horrific type of treatment for alleged mental health conditions or disorders or psychoses until the mid-1900s or so. So you can watch this video and learn a little bit more about William Scoville as a prolific lobotomist. And you can read more in the book if you would like to do so. And this is the article I would like you to read. So I'll post the video. This is where you can see how the cryoslicing of the brain tissue was done. And then you can read a little bit about what happened to HM's brain after he died. So this is assigned reading. It will be examinable. And I am going to give you a bonus discussion related to it. So you can watch the video, you can read the article, and then we'll have a discussion on Canvas. So I'd like you to tell me what you think about all of this. Read a little bit about HM, and let's talk about what we learned from him, and let's talk about some of the ethics that were involved before his surgery, after his surgery, and certainly after his death. So I've given you some talking points here. I've asked you what I would like you to do, and in return, if you do it well, this is worth up to a 1% bonus towards your final grade. So you've got almost a week, and sorry, this date down here is wrong. The date up here is right. You have until April 5th. So that's almost a week to do this. And I'm not asking for a lot, but 200, 250 words. Let's talk about what we think about all of this. Okay, so my gift to you, bonus 1%, if you do your part and summarize your well-informed, educated, researched thoughts in clear, concise, grammatically correct English. All right, so let's talk about this on Canvas and see what interesting things come up. I've got some summary questions. I will leave them for you to work through on your own, and I will have the next lecture up for you tomorrow. Stay well until then, and I'll talk to you on Wednesday.